Good morning, everyone, and thank you for being here today. I'm excited to kick off this presentation on airborne microbial contamination, specifically from a regulatory perspective. My name is Dr. S.K. Singh, and I hold a PhD in this field. In the next few minutes, we'll explore key aspects of airborne microbial contamination, its impact, and what regulations are in place to manage it. So, let's get started and delve into this important topic. Welcome to our discussion on airborne microbial contamination. This refers to the presence of viable microorganisms such as bacteria, fungi, and spores floating in the air that can potentially cause issues. This type of contamination is especially critical in environments like sterile pharmaceuticals, where maintaining purity is essential, and in biotechnology settings. Hospitals, particularly in operating theaters and ICUs, are also high-risk areas because the air quality directly impacts patient safety. The major risk here is twofold. First, contamination of the product itself, which can compromise its safety and efficacy, and second, the safety of the patients receiving these treatments. For example, during aseptic filling processes, if contamination occurs, it can lead to batch rejection, meaning the entire batch might have to be discarded, which is costly and delays treatment. So, controlling airborne microbial contamination is vital across these sectors to ensure product integrity and protect patients' health. Let's talk about the main sources of airborne microbes in our environment. First, personnel are a significant contributor, shedding microbes through skin flakes and respiration. In fact, each person can release around 1 million particles per day, making us a major source of contamination. Next, HVAC systems can also introduce or spread microbes if not properly maintained. Raw materials used in manufacturing or processing can carry microbes into the environment as well. Equipment surfaces are another source, as microbes can persist on these surfaces and become airborne through routine operations. Lastly, outdoor air infiltration plays a role, as microbes from the outside can enter the indoor environment, especially if ventilation isn't properly controlled. Understanding these sources helps us target our efforts to reduce microbial contamination effectively. Now, let's take a closer look at the different types of airborne microorganisms that we might encounter. First, we have bacteria, such as Staphylococcus aureus and Bacillus species. These bacteria can become airborne and pose a risk of contamination, especially in healthcare settings. Moving on, we also have fungi, including Aspergillus and Penicillium. These fungi produce spores that can easily become airborne and spread through the air. Additionally, yeasts are another type of microorganism that can be found in the air, although they are generally less resistant. Speaking of resistance, spores are particularly notable because they are highly resistant to environmental stresses. This means they can survive harsh conditions and remain a threat for longer periods. To illustrate this, consider Aspergillus spores in HVAC systems. These spores can travel through the ventilation system and contaminate sterile areas, such as operating rooms or clean laboratories. This example highlights how airborne fungi, especially spores, can be a significant source of contamination if not properly controlled. Understanding these different types helps us grasp the importance of air quality management in preventing microbial spread and maintaining a safe environment. Let's take a closer look at cleanroom classification according to ISO 14644. This system is based on the number of particles per cubic meter of air. Essentially, it helps us define how clean or controlled the environment needs to be for different applications. There are ISO classes ranging from 1 to 9, with ISO 1 being the cleanest and ISO 9 the least. In pharmaceutical settings, you'll often see ISO 5 environments designated as critical zones. These are areas where the highest level of cleanliness is required, especially when handling sensitive products or processes. Meanwhile, ISO 7 and 8 are more common for background areas, spaces that support the critical zones but don't require the same ultra-clean standards. For example, ISO 5 limits are approximately 3,520 particles of 0.5 micrometers or larger per cubic meter of air. This gives you a sense of just how strict the standards are in these critical zones. Understanding these classifications helps us ensure product safety and regulatory compliance. It connects directly to our overall goal of maintaining a controlled environment that safeguards the integrity of pharmaceutical products. Let's take a closer look at microbial limits as outlined by the WHO and EU GMP standards. These limits specify the maximum allowable levels of microbial contamination, measured in CFU or colony-forming units. Essentially, CFU is a way to quantify the number of viable bacteria or fungi present in a sample. When we talk about GMP cleanroom grades, ranking from A to D, each grade has its own specific microbial limits. Grade A, for instance, is the highest level of cleanliness, used in critical areas like aseptic filling. As we move down to grades B, C, and D, the allowable microbial limits increase accordingly, reflecting the different levels of environmental control needed for each area. Understanding these limits is crucial because they directly impact the safety and quality of pharmaceutical products. Maintaining these microbial limits ensures we minimize contamination risks, protect patient health, and comply with regulatory standards. So, in essence, these microbial limits serve as a benchmark for cleanliness, guiding us in designing and maintaining our cleanrooms to meet the highest standards of pharmaceutical manufacturing. In this slide, we're focusing on monitoring methods, specifically environmental monitoring, or EM. EM is essential to ensure the cleanliness and sterility of our pharmaceutical environments. We use several methods here. Active air sampling measures colony-forming units per cubic meter, or CFU-M3, giving us a quantitative idea of microbial presence in the air. Settle plates are a passive method, where plates are left exposed to collect microbes settling from the air over time. Surface sampling helps us check the cleanliness of various surfaces within the environment. Personnel monitoring is also crucial, as it helps us assess the microbial load carried by staff, ensuring they are not introducing contamination. It's important to remember that to stay compliant, we need to combine these methods. No single technique is sufficient on its own. Moving on to Indian regulatory requirements, we must adhere to the Indian Pharmacopoeia and CDSCO, or Schedule M guidelines, which set the standards for pharmaceutical manufacturing practices. Some key requirements include having a separate air handling unit, or AHU, for sterile areas to prevent cross-contamination. Continuous operation of HVAC systems is vital to maintain controlled environments. Finally, particle and microbial monitoring are ongoing activities to verify the environment remains within acceptable limits. All these elements work together to ensure our manufacturing environment remains compliant, safe, and sterile, aligning with overall quality standards. Now, let's take a look at some common sampling techniques used in our field. First, we have impaction samplers. These devices work by drawing air and forcing particles to impact onto a collection surface, which we then analyze. Next are filtration methods, where air is passed through a filter that traps particles, allowing us to assess the contamination levels. Lastly, we have centrifugal samplers, which use centrifugal force to separate particles from the air sample for collection and analysis. For example, we might take a 1,000-liter air sample, incubate it, and then count the colony-forming units, or CFUs, to determine microbial presence. It's important to note that these methods aren't strictly fixed by ISO standards. Instead, they are often defined by the user based on specific needs. When choosing a sampling technique, we base our decision on factors like historical data, what we've observed in past samples, and the process risk, meaning how critical the process is and the potential impact of contamination. If a sample exceeds predefined limits, it triggers an investigation followed by corrective and preventive actions, or CAPA, to address the issue. Finally, we set alert and action limits to help us identify when intervention is necessary, ensuring we maintain control and quality throughout the process. Let's take a closer look at the contamination control strategy, or CCS, as outlined in the latest EU GMP Annex 1. This strategy is fundamentally risk-based, meaning we focus on identifying and mitigating potential contamination risks throughout the process. It includes several key elements. First is facility design, creating spaces that minimize contamination risks from the start. Next, HVAC control is critical. Maintaining proper airflow and filtration helps keep the environment sterile. Cleaning and disinfection are also vital. Regular, validated procedures ensure that surfaces and equipment stay free of contaminants. And of course, personnel behavior plays a crucial role. Training staff to follow good practices reduces the chance of introducing contamination. For example, one of the most important design features in cleanrooms is unidirectional airflow, or laminar flow, especially in Grade A areas. This means air flows in a single, unidirectional stream, which helps carry away particles and contaminants, maintaining a sterile environment. All these elements work together within the CCS to ensure we control contamination effectively, supporting product quality and patient safety. Now, let's talk about some key control measures that help us maintain a sterile environment. First, HEPA filters are crucial. They can capture at least 99.97% of airborne particles, ensuring that the air remains as clean as possible. Next, pressure differentials play a vital role. Maintaining a pressure difference of about 10 to 15 pascals helps prevent contaminated air from flowing into critical areas. We also implement a specific number of air changes per hour, which keeps the air fresh and reduces the risk of contamination buildup. Gowning procedures are equally important. They act as a barrier to prevent contaminants from entering the clean area through clothing. Lastly, regular cleaning and sanitization are essential to eliminate any residual microbes and maintain overall hygiene. All these measures work together to create a controlled environment, supporting the overall goal of safety and sterility in our processes. Let's take a moment to look at the references that underpin the standards and guidelines we've discussed today. First, we have the WHO Technical Report Series, which provides global guidance on pharmaceutical quality and safety. Next is the EU GMP Annex 1 from 2022, a crucial document outlining good manufacturing practices specifically for sterile medicinal products in Europe. Then, ISO 14644 and ISO 14698 are international standards that focus on cleanroom classification and biocontamination control, key for maintaining controlled environments. The Indian Pharmacopoeia offers comprehensive guidelines on pharmaceutical quality standards specific to India, ensuring consistency and safety. Lastly, the FDA aseptic guidance serves as a vital reference for sterile manufacturing practices in the United States. These references collectively highlight the importance of adhering to international and regional standards to ensure product quality, safety, and compliance throughout the manufacturing process. For further support, please don't hesitate to reach out. We're here to help and ensure you have all the resources you need. If you have any questions or need additional information, feel free to contact us. And with that, I want to thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're committed to supporting you every step of the way. Thank you once again.